Good afternoon, everybody. I'm going to present our framework BioASF, which can be used to generate simulation software for simulating biological pathway models. This is a representation of a biological pathway model. A pathway is defined as a series of molecular interactions leading to a function or a change in a cell. Please note that a pathway can trigger or activate other pathways. So in order to save money and time and properly understand the working of complex biological pathways, they can be simulated. Let's see how simulation frameworks do their job. Generally speaking, a simulation framework provides a language in which a biological pathway can be specified. They then usually represent the specified pathway as a network, and thereafter they take care for information flow from one node of the network to the other one in order to realize a simulation. So these three characteristics, model specification, model representation, and model execution, are very important for a simulation framework. Let's see what the proper requirements should be with respect to these three characteristics for a simulation framework. Regarding model specification, it is not desirable to have a weak typed language. Why? Because in this way, the nodes of the network representation of the models specified in these weak typed languages are of very generic types. And therefore, it is possible to almost connect every node to the other one without having any biological meaning. Therefore, it is desirable to have a strong typed specification language which provides distinguished biological types such as catalysis, complex assembly, protein, and etc. Why? Because in this way, it is easier to check the validity of the models and write models which are less error prone. Regarding model representation, again, it is not desirable to have a flat network structure. Why? Because in this case, it is not possible to define a node as a separate sub-pathway which can be referred from main pathway. So we prefer to have a hierarchical network. Why? Because it is then possible to reuse and also integrate the models developed by other groups into your own model. Regarding model execution, it is not desirable to have a centralized decision-making at network level. Why? Because in this case, a big monolithic network interpreter decides which action should be taken, by which node, and when. This makes simulating complex biological pathways more complicated. So it is desirable to have a decentralized decision-making at node level, with each node having its own local rules. So this is very important. Each node has its own local rules. This way, it is possible to simulate large biological networks, and simulating of large biological networks become more maintainable and also scalable. Unfortunately, the current simulation frameworks don't satisfy all these requirements. Therefore, we come up with a new simulation frameworks which satisfy these requirements. The question here is, should we start from scratch, or should we look at around us to see whether there are existing technology which can be used to satisfy these requirements? We did. We noticed that BioPAX can partly satisfy our requirements regarding model specification and model representation. We also found out that the existing agent technology from the artificial intelligence discipline can help us a lot. So BioPAX not only solves the problem of diversity of data formats and visualizations, but also it supports hierarchical pathways, and it provides handy tools and software libraries for model developers. Additionally, nowadays, major pathway databases support BioPAX. So let's see what BioPAX is. BioPAX is a semantic web-based language in which a network of biological entities can be specified. BioPAX has three main core concepts, which are interaction, pathway, and physical entities. And if we look at interaction, it has subtypes of control interactions and conversion interactions, and they are also they have their own subtypes, and these subtypes are organized in a hierarchical fashion. Now, BioPAX is a very great language to specify your biological pathways, but it doesn't provide any simulation execution semantics. And therefore, we come up with our framework, which is called BioASF. BioASF inputs are a biological pathway model specified in BioPAX, initial concentrations of physical entities, rules governing when a simulation, when a biological interaction should take place, which we call simulation rules, and also rules governing what to do with the results of simulation. So BioASF has a lot of attention to separation of concerns. And the output of BioASF is a multi-agent system which is ready to run. Now, the idea behind BioASF is actually very simple. Associate a software agent, a software robot to each BioPAX interaction and the BioPAX pathway, and let these agents communicate to each other in order to realize complex simulations. This is an example of a pathway specified in BioPAX. What we see here are BioPAX pathway concept, that big box. And we see also BioPAX interaction concepts such as complex assembly, template reaction, and we see also BioPAX physical entities such as CTNMB1, AXIN, and we also see here are BioPAX properties which connect the BioPAX concepts to each other with certain restrictions. So what you see here is CTNMB1 and TCF-left are left participants of the complex assembly interaction, which produces a complex which plays the role of a transcription factor for the transcription of the AXIN protein. Thereafter, AXIN is transported from the nuclear to the cytoplasm, where it forms destruction complex together with other proteins. So here comes, this is very important. This actually, BioASF adopts this BioPAX network structure and adds about that a network of agents which cooperate and communicate with each other to realize a simulation. So let's see how different agents within BioASF communicate with each other to realize a simulation. Simulation starts with analysis agent, which from one side, from one hand, it activates environment agent, and from the other hand, it activates top-level pathway agent. Pathway agents recursively activate sub-pathway agents and their interaction agents. Interaction agents, upon their activation, wait to receive changes in the concentration of their physical entities from environment agent, and environment agent, upon its activation, loads initial concentrations from a repository and propagate them to interaction agents. And then when an interaction agent receives concentration of its physical entity based on its local rules, decides whether or not to perform an interaction, and consequently consumes physical entities or produces new physical entities, which goes back to environment and causes another feedback loop. So in order to validate applicability of our framework, we simulated two types of networks. A signal transduction network, which modeling Wnt beta-catenin signaling pathway, and a gene regulatory network modeling hematopoiesis stem cell regulators. In both studies, we converted the original models, which were written in a proprietary language, to BioPAX, and we provided simulation rules and analysis rules, and we were able to reproduce the results of the original experiments. Regarding Wnt beta-catenin experiment, we calculated beta-catenin levels for the initial concentrations of Wnt from 0 to 5 over 100 steps. And we got almost the same result as the original experiment. And for gene regulatory network experiment, we built a large network in which each node represents a cell state consisting of 11 genes. And within this big network, we found terminally strong connected components, TSCC, 3 TSCCs. A TSCC is a subnetwork of a network in which every vertex is reachable by other vertex. And there are no outgoing arcs to the outside of the subnetwork. And it is believed to represent the cell's stable state. So the first TSCC represent a known hematopoiesis cell. The second one, consisting of one node, represents a mature erythrocyte. And we have also TSCC3, which consists of 32 nodes, and they together represent heterogeneous hematopoiesis cells. So in summary, BioASF together with BioPAX promote model reusability by supporting hierarchical networks. They facilitate model validity by providing strong type specification language. They provide scalable model execution by using agent technology, which is inherently decentralized, and they increase efficiency of simulation development by generating simulation software instead of writing a simulation software for a specific pathway. Now, finally, I would like to thank my co-authors for their valuable contribution, and ISCB for funding my travel. Thank you very much.